Good afternoon, folks. We are back at the EOCP's annual conference in the water stream. We're continuing on this afternoon with a presentation by Shawn Dubois from IDEXX Laboratories. Shawn is giving us a lecture on enzymatic methods for detection of bacterial indicators of water quality, specifically total coliforms and E. coli. Now, Shawn is uniquely qualified to give this chat to us today because he's a senior scientist with IDEXX Laboratories. He's been a part of the water industry for over 20 years, working in R&D, and in his current role, managing the product support group for North America. In addition, Shawn also manages the ISO 17025 accreditation for water quality control labs. Shawn has a bachelor in biology, a bachelor in environmental science. He's also a participant in several standards development organization, including standard methods for the examination of water and wastewater. So I am so glad you're here to talk about this, Shawn.
Thank you, everybody, for joining me today, both here in person and virtually. So, as mentioned, I'm going to go over enzymatic methods for detection of coliforms and E. coli and water. So, I'm going to start off going a little bit over an introduction to microbiology, again, then go over some of the enzymatic methods, spend a little time on QA and QC, and then have some time at the end for questions. And actually, I'll stop a couple times during the presentation too and see if anybody has any questions that they want to ask. All right, so again, start off a little bit of an introduction on microbiology. Unlike poor Roger here, there won't actually be a test. This is purely just for your knowledge and also just kind of help understand when it comes to testing water, why we do it and what we're looking for. So always like this is a picture of my daughter waiting to go into a kayak, into a lake, but she has to wait for a family of ducks who are decided to come walking by as she's about to try to get in. And kind of, this is a great little picture to show when we talk about the bacteria that we're looking for contaminated waters, kind of where they come from. The bacteria we're looking for, they can come from naturally occurring in the water, they can be occurring naturally in the soil, they're going to come from animals and, of course, humans. So everything we're looking for is going to come from one of these kind of locations. To kind of start off, figuring out how bacteria grow is really important to how any of the tests that we're going to talk about work and how they will tell us whether or not the bacteria are present. So when we talk about drinking water or any kind of treated water, bacteria has been gone through some kind of treatment process, right? So the bacteria has been, in a sense, injured. It's not healthy anymore. We kind of hope that we kill most of it off. But the truth is it's still surviving. It's just not very happy. And so what we're going to do is we're going to take that bacteria and put it into an environment that's enriched with nutrients and other materials that are going to help the bacteria recover. And then at some point, that bacteria is going to start growing. So there are different phases in kind of a general life cycle of bacteria once it's put into a nice, happy environment. The first phase is the lag phase. This is a time when the bacteria is basically taking in a bunch of nutrients, it's making itself healthy, it's growing, it's beginning to begin cellular division. Next part is the log phase. This is where the bacteria are actually starting to divide. So that population is starting to double at every certain time point. So if we start off with one bacteria, that bacteria is going to become 2. Those 2 become 4, 8, 16, 32, 64, 128, and basically the population just continues to double. And at this point, this is kind of where we're going to start seeing our indication that that bacteria is present in the sample. And this, when we talk about testing, lag phase and log phase are really important. And I'll kind of refer back to these later on in the presentation. The third phase is the stationary phase. This is kind of a point where the population is neither increasing or decreasing anymore. Some bacteria are growing, other bacteria are dying. So the population is kind of just staying at a standstill. And then finally, we reach the death phase. At this point, the population is shrinking more than it's growing. But again, for us, when we talk about testing, what we're concerned most about is the lag phase and the log phase. So after that very general introduction to how bacteria work, now talking about like when we actually are looking for bacteria, why we're looking for them and what we're looking for. So when we're testing water, whether it be drinking water, wastewater, recreational waters, source waters, what we're looking for is something that's going to tell us whether or not the water is safe. So things that can make the water unhealthy could be different viruses, different bacteria, different organisms. And there's just way too many types. There are way too many different ones that are out there. And to look for them all is very difficult, time consuming, can be expensive. It could actually be dangerous for the lab personnel. So rather than spend all that time, energy, money looking for anything that could be in there, we look for something that will indicate that the water has been contaminated. So the best kind of indicator organisms are ones that are going to be present when pathogens are present in the water. They'd be absent when pathogens are not. We want something that's going to be in very high numbers, so that way we can take a very small sample, and within that small sample, determine whether or not the water is safe. We want them to be able to survive as well or better than the pathogens in the water, so that way we can find them. We don't want something that has a short life, so they disappear before we have a chance to find them. And then when it comes to the actual testing part, we want something that's going to be easy and safe for people to test for. We don't want to put anybody at risk. We want to have results pretty quickly. We want to be able to make decisions fast. We want something that's relatively inexpensive. And really above all, we want something that's accurate. We want to be able to say with confidence that when we say water is clean and safe, that it is truly clean and safe. And if there is something wrong with it, we want to be able to find it and then take the appropriate action to make sure that we don't put anybody in jeopardy. So, any questions at this point? Cool. Okay. So when we talk about indicator organisms, the ones that we tend to hear about the most are coliforms. So coliforms are a group of bacteria that have certain characteristics, and they all belong to the family Enterobacteriaceae, for those who maybe remember their high school biology class. But basically, there are different genus that fall within this family that kind of fall into that coliform group. And not everybody here in these groups are coliforms, but certain ones. And they kind of fall into two different types of sources. Again, going back to that picture of my daughter, there are ones that occur in animals, including humans. And this includes E. coli, different species of Enterobacter, some Klebsiellas, Citrobacters, and so on. But this also includes bacteria that fall, that can be found naturally in the environment. Again, you have some Enterobacters, Klebsiellas, Serratia. So again, coliforms, group of bacteria that can be coming from both an animal source, human source, or an environmental source. So coliforms, there's one that's particularly important to us, and that of course is E. coli. So whenever we talk about kind of the best indicator for fecal contamination, E. coli is the one that we focus on. So E. coli is a major inhabitant of the intestinal tract of humans, warm-blooded animals, but really humans as well. It's got a very high number of them in us. There's just as many of them in us as there are on the planet. They're shed in very high numbers. They survive very well when they're in the environment. And in general, they do not naturally occur in the environment. If we are finding them in environmental waters or in ground or any place like that, that's because there's been some kind of fecal contamination. So they are basically a true indicator for fecal contamination. Some other bits, they're gram-negative, which is just the type of bacteria that they are. They're facultative anaerobes, which basically means that they can survive where there's oxygen or where there's no oxygen. And they're also thermotolerant. So they're able to survive in a wide range of temperatures, including temperatures above body temperature. So they're great bacteria. They grow very well. They're quick to grow, and they can survive in most environments. So, any questions at this point? Cool. All right. Now actually getting to the test methods. So we know what we're looking for. We're going to look for coliforms. We're going to look for E. coli, and how do we do so? So how many people here are actually doing testing in the lab? Okay, quite a few. So there are different types of testing that can be done. Kind of the way everything's kind of started off was with multiple tube fermentation. Multiple tube fermentation is kind of a lengthy process. There's a lot of steps. It's up to 60 steps total to run the test. Not exactly quick. Results take up to 4 days for confirmed results. And there's a lot of points where things can go wrong. And so accuracy is kind of low when it comes to this kind of testing. So not the best, but you know, it tells you something. Next comes membrane filtration, which is an improvement over multiple tube. Definitely a reduction in the number of steps. It's about 20 total from start to finish. And results are between 24 and 48 hours, depending on if the test has a confirmation. So definitely faster, more accurate, and easier to do from a number of steps standpoint. However, there's still a lot of technique that's involved with membrane filtration. And with that, there's opportunities to mess it up and therefore not get exactly the best results for your test. And then finally, we come to enzymatic methods. And these methods are much faster. It's all about a total of 8 steps for quantification, less if you're doing just presence absence. And results are confirmed in either 18 or 24 hours, depending on which test we're looking at. So when it comes to the enzymatic methods, basically we have a media, and that media is what's going to be able to tell us whether or not that bacteria is present. So there's kind of like 3 groupings of ingredients that can be found in the different reagents. And these include nutrients, supplements, and other things that are present in order to help that bacteria during that lag phase, in order to help them recover, become healthy, and begin to go through that process of division. So there's a bunch of that in these reagents. Next, we have inhibitors, and these are there to prevent any kind of other bacteria or other organisms that might be in that water from interfering with the test and preventing it from working correctly. And then finally, we have the nutrient indicator. And this is the part of the reagent that is going to be able to tell us whether or not our target bacteria are present in the sample. So first test I'm going to talk about is COLR. So COLR is used for the detection of coliforms and E. coli. It is a 24-hour test. Now, I'm from the main office in Maine, Westbrook, Maine, in the States. So I apologize. Most of my stuff is going to say EPA on it because that's where most of our focus is. But all these tests are also approved in the different provinces of Canada. COLR is also found in standard methods for the examination of drinking water and wastewater. It is in section 9223B. Again, we have approvals for many different types of waters depending on where you are. And COLR is one of the most approved methods around the planet. COLR 18 is a very similar test. Only difference here is that it is an 18-hour test, so we get results 6 hours faster. Again, approvals, including in standard methods 9223B. COLR 18 is also an ISO standard. So if you are in an area or a country that looks at ISO for your testing methods, COLR 18 is an ISO standard, 9308-2. And again, can be used for many different types of waters. So again, all these reagents have similar nutrients, inhibitors in order to let our bacteria grow and prevent other ones from interfering, and then the nutrient indicators. So for COLR and COLR 18, there's actually 2 different nutrient indicators. The first one I want to talk about is the one to detect for coliforms. So this is O-nitrophenyl beta-D galactopyranoside, which is way too many words. So ONPG, nice and easy. So ONPG is basically got two components. There's the first one, which is the nutrient, which in this case is the beta-D-galactoperoxide. And then bound to that is the actual indicator, and the indicator is O-nitrophenol. So these two parts are combined together, and when they are bound, the appearance of them is colorless, so you don't see any color. Now what we're doing is we're looking for a specific enzyme that coliforms have, and that enzyme is beta-galactosidase. So if we have a coliform in our sample and we've added choline or choline 18 to it, coliform is going to take in the ONPG, brings it into the cell, and then if this enzyme is present, that enzyme is going to bind to the ONPG. And when that happens, the bond between the nutrient and the indicator is broken, and that indicator is now floating freely, and it will actually turn yellow. So for choline and choline 18, ONPG is going to tell us coliforms are present. We're going to see the media go from colorless to yellow. The second indicator is used looking for specifically E. coli. And again, E. coli is a coliform, so it's able to, has a beta-galactosidase, so it's able to utilize the ONPG. It has another enzyme that's pretty unique to E. coli, which is this beta-glucuronidase. So we have another nutrient indicator looking for this enzyme. In this case, it's 4-methylbifferyl beta-D-glucuronide, which I have to say, after 21 years of working at IDEXX, I finally get used to saying this without having to read it letter for letter. But again, we like our acronyms, and this acronym is MUG. So again, keep it easy. But it's the same concept where we have a nutrient, and bound to that is an indicator. In this case, that indicator is 4-MU. When these are bound, and if you look under a UV light, there'll be nothing to see. Now, if E. coli is present, and this enzyme is being utilized, again, it will bind to that nutrient, breaks that bond, the indicator is released, and it will now fluoresce and glow when under a UV light. So for choline and choline 18, yellow tells you coliforms, yellow with fluorescence will tell you E. coli is present. Any questions on those? No? Okay. So our third enzymatic test for coliforms and E. coli is ColiSure. ColiSure is a very similar product. It's a 24-hour test again. Again, same standard methods, still same similar approvals. The difference with this product is the nutrient indicator that's used to look for coliforms. Whereas choline and choline 18 use ONPG, for ColiSure, we're using CPRG. And the only difference here is the indicator that's attached. We're still looking for the same enzyme. We're still looking for that same nutrient usage. The difference is that indicator. And this indicator is chlorophenol red. So when this product, when this nutrient and indicator bound together, the appearance is actually yellow. So ColiSure starts off yellow. Coliforms are present, utilize that nutrient, bond is broken, that indicator is released, and it will turn red. So ColiSure turns red for the presence of coliforms. And again, it contains MUG as a second indicator to look for E. coli, works the same way. So that's basically the simple way of looking at the science of how these enzymatic tests work. Now, moving on, these tests can be both run as a presence-absence test and as a quantification method. So it's either a yes, no, or a number. And I know depending on where you're located, you might be able to do one or the other. The important part is that it's the same products, the only difference is in how you process that sample. So if you're doing presence-absence, it's pretty simple. If you ever use like flavoring to add to your water, then you can use our test, because that's exactly what you're doing. You're basically taking that powder reagent, snapping it open, adding it to your 100 ml sample, mixing it to dissolve it, and then you incubate it. And then after the appropriate amount of time, you remove it and look to see what the results look like. And that's presence-absence testing. If you're doing quantification, there's one additional step. So basically in this case, you're going to add your reagent, and then you're going to use the quantification system, which includes the quantitrays, which are the actual quantification device for the test, rubber inserts, which are going to allow us to seal that sample, And then finally, the quantity tray sealer, which is going to actually take that quantity tray and make it usable. So the way this works, you start off the same way where you have your 100 ml sample, you add your appropriate reagent, dissolve it, pour it into the tray, which basically at this point is kind of like a bag. There's a plastic front with wells, and then the back has an adhesive layer with a foil paper combination. That gets put into that rubber insert, that gets put into the sealer. And basically what happens is that sealer is going to take that sample and kind of just act like a squeegee, push it up and fill that tray as it goes through. And at the same time, it's going to take that adhesive and make it seal off each of those wells individually. So it takes that 100 ml sample and turns into many multiple little tests. You then incubate that for the appropriate amount of time, then read results at the end of the incubation. Some kind of hints on using it is that with any tests that you do for microbiology, it's always about being aseptic. You want to make sure that you're not contaminating your sample. Same thing if you're collecting the sample and sending it someplace else. How you collect that sample is really important to make sure that you don't introduce any bacteria that would cause a false positive. When it comes to the tray, carefully, squeezing at the top, kind of make it open, and you pull back that foil tab, and then you easily pour in your sample. The tray is sterilized, so you want to make sure that you don't touch the inside, because then you can contaminate it, and again, you get bad results. Okay. So actually running the tests, if you're using Kohler and Kohler 18, the samples are run at 35 degrees C for incubation. If you're using Kohler 18 as a presence-absence test, so if you're just keeping it in that 100 ml volume, there is a pre-warming step that's required in order to make sure that's the appropriate temperature. So if you're doing PA testing, you have to pre-warm that sample for a certain amount of time in a water bath, depending if it's at the appropriate temp. And then you incubate it for the remainder of the 18 hours. If you're using the quantity tray format, there's no pre-warming. You just add your Kohler 18, seal, and then incubate. So Kohler is a 24-hour test with results valid up to 24 hours. So it gives you a 4-hour window in order to read your results. And then Kohler 18 is an 18-hour test with results valid up to 22 hours. So again, presence absence, very simple. Color, yes, no. If you do have yellow, that's a positive for coliforms. Again, you're seeing that usage of that ONPG. And then if you do have yellow, you would then look under a UV light, 365 nanometer, and see if you also have fluorescence. Yellow with fluorescence is a positive for E. coli. Quantification is the same thing. Only difference here is that instead of counting like a yes or no, you're actually counting individual wells. So if you have wells that are yellow, you then look under a UV light and see if those yellow wells also fluoresce, and then you know how much E. coli you have. Now, with any test, there can be some kind of variation in intensity of signals. Sometimes you might have weak to really strong yellows, and the same thing with fluorescence. So as an example of a Kohler or Kohler 18 tray, you can see this well is very dark. So it's yellow if you look under an ambient light, but there's no fluorescence. It's coliform positive, negative for E. coli. Then you have this well, which has a nice bright, strong fluorescence, positive for E. coli, because it has yellow and fluorescence. And then you have this well, which kind of has more of a muted fluorescence, but it's still present. So this is still a positive for both coliforms and for E. coli. Now, if there's any kind of question of whether or not something is positive, there are comparators available for Kohler and Kohler 18. And basically, it will tell you the minimal amount of yellow or fluorescence in order to call a sample positive. So if there's any kind of question, you can kind of use a comparator, and it'll help you decide whether or not to call it a positive. And it comes for both the quantity tray formats and for presence absence vessels. Okay. Colisure, again, coliforms and E. coli, we're looking at incubating samples at 35 degrees C. And again, colisure is a little special, is that it's a 24-hour test with results valid up to 48 hours. So it gives you either a day or 2 days in order to be able to read your results. But again, we're looking for that usage of in this case, the CPRG. So you're looking to see if sample turns from yellow to red magenta. If If you see that, then you have a positive for coliform. Then you put under a UV light and look to see it awful fluoresces. Again, same thing for quinitri. So again, just like with any test method out there, you can have kind of a variation in signal intensity. So with coliforms, as long as the well or the vessel is a red or magenta color, then that'd be considered positive. If you see something like an orange, then that wouldn't be considered positive. You have to incubate a little bit longer or be a negative. The same thing for fluorescence is that you have no fluorescence, positive for coliform, negative for E. coli, strong fluorescence, positive for E. coli. And then there's this one up here, which is a very dull fluorescence. That's still magenta and there's still some fluorescence. It's considered a positive for E. coli. Unlike Kohler and Kohler 18, there is no comparator. So basically any color change from red magenta is a positive for coliforms, and any wells that also fluoresce, no matter what the signal intensity, is a positive for E. coli. Any questions at this point? No? Okay. So presence absence is easy. Yes or no? Then you're done. For quantification, there is a next step where after you count the number of positive wells, you then refer to a most probable number table to tell you what your final result is for that sample. So if you're using, there's two different tray types. There is a 51 well tray, basically has one size well. There's 50 of one size, and the top well is the 51st. You basically have a chart that has a few columns. The first column, 0 to 51 wells, find that number, move over to the next column, and that's your result. If you're using the EOtec2023, it's a little bit different. There's actually two different size wells. There's 48 small and 49 large. 48 small ones of the same size, and then the 49 large, there's 48 of one size, and the 49th is the top one. For the EOtec2023, you have to count both wells individually. So you count the number of positive wells that are large, record the number of positive wells that are small, and then basically use this our little handy eye chart. And first column is number of large positive wells, and then the top row is the number of small positive wells. And basically what you're doing is you're going to take those two numbers and see where they meet. And where they meet is your result for that sample. So example, if you had 48 large and 25 small, your most probable number for that sample would be 344. So the advantage of the quinitri system over membrane filtration is that there's a much larger counting range. And because of that counting range, you don't need to do dilutions as often. So hopefully in drinking water, there's never an issue with that. But if you're on the wastewater side or didn't waters that you weren't sure what number you would have, it is possible to do dilutions with the enzymatic methods. The most important part to remember is to always dilute your sample before you add the reagent. It seems common sense to do that, but we've had plenty of times where people have diluted out the reagent and they can't figure out why their test doesn't work at all. So always important, dilution first, add your reagent, and then basically continue running the sample as you normally would. After you record your result, it's just important to remember to account for whatever dilution that you ran in order to get your correct final test result. Okay, any questions? Nope. Okay. Yeah, switch gears a little bit and talk about TECTA briefly. So TECTA is a new test to IDEXX. It's for the presence absence testing for coliforms and E. coli, again in water. So works on a very similar principle to Kohler and Kohler 18. The difference here is that this is an automated test. So you still have a reagent that's detecting the products. Difference here is that there's actually a machine that's involved that will actually tell you whether or not the sample is positive. This is approved by Health Canada and EPA for presence absence of drinking water. Very simple to run. You basically have a cartridge, a vessel, that contains the reagent already in it. You pour in 100 mLs of your sample, swirl to dissolve, and then you enter information into the machine about what your sample is, the type of water it is, what you're looking for. And then you add the cartridge into the actual Tecta device, close it, and what this will actually do is it's a self-contained unit that will incubate the sample. And during that time, what it's also doing is reading the sample and seeing whether or not it turns positive continuously. So it works under the same principle of other enzymatic methods. What we're doing here is we're just changing the indicator that we're using. So we're still using the same nutrient. We're still looking for that same enzyme that's present in coliforms and E. coli. The only difference is that we're changing the indicator. So this is an example of what the one is for for E. coli. But we're doing the same thing. Basically, if that enzyme is present, that bond will be broken, the indicator is released. And in this case, both indicator for coliform and E. coli is fluorescent. And it will turn hydrophobic, which means it does not want to be in water. And what it'll actually do is it will move into the bottom of that plastic cartridge, and it will collect into one spot. And what the machine does is it actually looks for fluorescence that occurs in that spot. And once it reaches a specific value and sees that change, it will trigger it, and then it will say whether or not this sample is positive for coliforms and E. coli. And then the system can either just maintain that information on itself. It can also be set up to send out an email. So if you are not present with the machine, you'll still get the results coming to you immediately as soon as it has a final answer. For the coliform E. coli test, it is an 18-hour test, which means that if the sample is negative, it'll take 18 hours to tell you that. But if is there anything in there, it will turn positive sooner than that, and it'll send you that message if you have set up for that. So, any questions on Tecta? No? Okay. So moving on to quality control, which is always an important step when having any kind of product. You always want to ensure that the test is going to perform as intended, that it's always going to give you the result that you expect. So we always suggest to use a pure culture strain, something that is going to give you a definitive result. So always getting a strain from a natural collection such as ATCC or NCTC, so that way you know that the bacteria is performing as expected. IDEXX also does have a QC product line that can help you with this too. It's one-time use. It'll give you your target bacteria that you're looking for, whether it be E. coli, coliforms, and then a non-target bacteria to make sure that that won't turn the test positive. And then we also have other tests that are available, QCs that are available for our other product lines that we run. And then finally, talking about certificates of analysis. This is very important for any of your records. If you're ever audited, especially if you are a lab that has accreditations. These can all be found on our website for all of our products. And basically what it goes through is it tells you all the different testing that was done on that lot to make sure that it was good to use before we would actually release it for distribution. We have a very stringent policy of how our products get through from manufacturing to our customers, because we want to make sure that everything that you get in your hands is going to work as intended. So all of our products, reagents, are manufactured in Westbrook, Maine. All those materials are collected when they build an entire lot. We collect a sample from across the entire lot as we're building it. That is then given to our quality control lab, who will then do battery testing. Once all that testing is complete, all the paperwork is filled out, that goes to our quality assurance team. They review to make sure everything has been completed and that everything is correct. And then after that, they will release that a CA certificate of analysis, and that allows that product to be released for use by our customers. So we have several steps that prevent anything from getting out into the field that would not be working as intended. And IDEXX as a whole, because I don't know if how many people here know IDEXX, but it's a relatively large company. We're not, the water business is actually probably the smallest part. We also have a division that works with livestock, poultry, and dairy. And then we have a much larger part that deals with companion animals. So if you have any pets and you take them to the vet, there's a high probability that there's an IDEXX test being used to make sure that your pet's being healthy. So our entire company has 9001 and 14001 certification with ISO. And then our water QC lab specifically has 17025 accreditation. And what this means is that twice a year, our lab is audited to make sure that our personnel, our equipment, our lab, and our entire processes are functioning as intended. It just gives an extra layer of security that we're releasing quality product. Not so much in North America, but in Europe, this is actually kind of big, and in Australia and New Zealand. And it actually kind of reduces some of the QC testing that labs have to do because we have this accreditation. But briefly, to get our C of A's, you just go on our website. There's a couple different ways you can look for certificate of analysis, find that, you type in the specific lot number, hit request certificate. That'll bring up links to different PDF files for your product. You click on that, and then it'll open up a PDF, and you can either save it or print it and have it for your records. And again, regardless of what product it is, whether it be one of the enzymatic methods or it's the quantity tray or our vessels or anything like that, that's all found on our website at that link. So with that, any final questions?
So with regards to the quantity trays, there's the 51 well tray, and then the other is
It's a quantity tray 2000.
2000. So
Yep.
what would be the use case for either, like, one over the other kind of thing?
Yep, so a lot of it has to do with the counting range. So the 51 well will give you a range of up from less than 1 up to 200. And the quantity 2000 is less than 1 up to 2419. So it's a much larger range. So especially if you're dealing with wastewater, it makes sure that you don't have to, lessens the chance you have to do a dilution because it has such a high range.
Perfect. Yeah, that's my question. Thanks.
Yep.
Hi, just wondering how far down the road we are to being able to purchase this and use it for reporting to our local health authority. Or is that in the works?
Yeah, so I am very disconnected at this point, truthfully, with regulations in Canada. However, we do have our government affairs team that works pretty closely. And if you want to, we have our booth, and we can get information from you and like exactly what you're looking for, and then we can reach out to that team and get that answer for you.
Yeah, I'm just, well, I guess if they're sending you here and looking to sell the product in Canada, I would assume that they're talking to Health Canada.
Yeah, so our products are approved for use. Again, I'm not sure on which specifically which waters we're talking about.
Oh, Fraser Health Authority for me.
Yeah. So again, like after, if we want to have a conversation, because Claire, who's our salesperson who's here, she has pretty good knowledge of what that is. But we are approved for many water types here in Canada as well for with these enzymatic methods like choline and colisure.
Thank you.
Any questions online, Kim? Okay. Any other questions, folks? No? Okay. Great. Well, thank you very much for coming and speaking with us today. That was absolutely super. If we can have a round of applause for our speaker, please. Thank you very much, Shawn. Yep. You did great. So I would just remind everybody to make sure you get scanned on the way out. And again, I look forward to seeing you at our final presentation for today in 15 minutes from now. And oh yeah, I forgot. Yes, present. So again, thank you very much for presenting. That's wonderful. All right, enjoy your break, people, and we'll see you shortly. Yeah, no, thank you for coming. That's just wonderful. And you know.